Hello, my name is Corynn Knight. My lab partner Kaylin LaFaro and I have been doing a project on the immune response to modified live vaccines versus killed BRD vaccines in weaning calves. So the purpose of this project has been to compare the efficiency of a modified live bovine respiratory disease, or BRD vaccine, against a killed BRD vaccine. This is a continuation of SBU's agricultural cattle study that began in 2019. And this project is specifically looking at whether the modified live vaccine is more efficient than a killed vaccine in a low-stress environment specifically. So there are a few differences between the two vaccines, killed versus modified live. And the killed vaccine is also considered something as an inactivated vaccine, one that contains no living forms of that germ or virus. It is also the most commonly used among ranchers, and also the simplest to use since it requires very little prep or storage. With our modified live vaccine, it contains a weakened form of the germ or the virus, so it has a partially living activated form of that virus, which is then able to cause a greater response within the immune system. It does require specialized handling though. It needs to be kept within a dark space or a very cool and dry space. And once activated, it is only viable for about 2 hours. This activation does take some intentionality. It takes some activity in that it requires mixing a dry form of the virus and a liquid medium, which is usually sterile water. So it does have that additional element to it as well. This is all being done within a low stress environment, which is basically part of the weaning method that we're using for these calves. Instead of just traditional weaning, which is where you separate the calf from the mother, we use what is called modified weaning, or a two-step method, in which we insert a nose flap, which is a plastic ring that goes within the nose of the calf and just kind of rests on the septum. And it doesn't puncture anything, so there's no physical harm, but it does keep the calf from being able to nurse. It doesn't keep it from being able to feed or drink in any other way, and allows it to remain with the mother. So overall, it decreases the stress that the calf experiences, which is why we call it a low stress environment. It has been found that when there is an increase of stress within the calf, there is an increase of the stress hormone called cortisol, which does cause a decrease in vaccine efficiency. BRD has many implications within the agricultural community. For the rancher, it has a very large financial impact. For every animal or cow that is affected, there is about a $100 cost to treat it. That may not seem like a lot just when you think about it, but if you start considering the number of cows that are part of each herd, it adds up very quickly. Within animal welfare, BRD is considered a multifactorial disease that's linked to pathogenic bacteria and viruses, and it lands in the respiratory system usually. It is the leading cause of morbidity and mortality of bovine populations. And even if this disease is caught, if it's not treated correctly or quickly, it can often lead to some severe permanent damage for the calf. One example of this would be what is called a lunger, in which an animal has experienced such tremendous lung damage that it causes the breathing to become difficult and to become very loud. And you can hear that breathing of the animal from about 50 yards away. So it's better to vaccinate for it and keep the calf from even contracting BRD than to even try to catch it and treat it. So from all of this, our hypothesis is that in a low stress environment, the modified live vaccine will show a greater immune response than the killed, allowing it to be a more efficient vaccine against BRD. For our experiment, we had 8 calves and we separated them up into 2 groups. Our first group received 4 calves that received the killed vaccine and a nose flap. And group 2 had 4 calves that received a modified live vaccine and the nose flap. The timeline for our blood sampling loosely followed the vaccination curve. in which we took our initial sampling to get a baseline and did our initial vaccination and applied the nose flaps all at time or day 0. And then on day 21, we did our booster vaccinations, but we also did our second set of sampling. And on day 21, we were able to document the highest number of antibodies that are going to be produced from that initial vaccination. Since by day 21 weaning is mostly completed, we also removed the nose flaps. Our final sampling occurred on day 45, which was able to catch the majority of the antibodies being produced from that initial vaccination as well as the booster vaccination, which gives us a good representation of the immune response of each animal. So our methods for this experiment, we've pretty much gone over the majority of the ones that were in the field looking at the timeline. But just to review, these vaccinations were administered to the 2 groups that I had previously mentioned. And all this was done at JMK Ranch, a local ranch in southwest Missouri. The samples were pulled on days 0, 21, and 45. And these blood samples were taken from the coccygeal vein of the calf, which is the lower portion of the tail near the body. They were loaded into red top tubes and put into a cooler to be transported back to the lab. And the nose flaps were removed on day 21, the same day that the booster vaccinations were administered. Within the lab, we centrifuged each sample twice, and this was done for purity. You can read in literature that it says to centrifuge it once, but we found that by centrifuging it twice, we are given a more pure sample, and therefore it's less contaminated by debris or other forms of the blood that we are not needing. This also allows for our serum, which is the portion that we use, to be separated from the formed elements, which are not necessary for this experiment. The serum was pulled off the top of each sample, transferred to a 1.5 microliter Eppendorfer tube, and then put in the freezer until all samples were collected. Once we had received the samples from all days of sampling, we thawed out each blood sample and were able to put it into the ELISA plate, which is shown here on the bottom, which allowed us to analyze each sample and test for the antibodies produced against the antigens in each BRD, for BRD in each sample, I'm sorry. It allowed us to be able to get our results and then to be able to compare the different groups. So for our results, we have our days of sampling along the x-axis, and we have the measure of the neutralizing antibodies along the y-axis. And since our antibodies are being measured via a proportion, the closer the number of antibodies gets to 1, the more antibodies are produced and the greater the antigens were neutralized within the blood sample. For our columns, you see that our killed vaccine antibody production is shown in light blue here, and our modified live antibody production is shown in dark green. We also see that on day 0 or time 0 here, there is very little antibody production, which is what we expect to see. But from then on out, you see a positive production of antibodies in both day 21 and 45, with it eventually leveling off on day 45 here. In each of these days, both of them, you do see that positive increase, which does tell us that you are receiving an immune response to the vaccine. But we specifically see on day 21 that there is that positive increase from in comparison to time 0, but there's also a significant difference between the 2 vaccines, with a greater concentration of antigens in our modified live compared to that of the killed vaccine. So in conclusion, from these results, we can determine that there is a large positive response from both vaccines in the low stress environment, meaning that both would be effective to prevent BRD. But we also see that there's that greater concentration of modified live antibodies on day 21, which suggests to us that we do need to do some further evaluation to see if this remains true, that modified live is more effective on day 21 than that of our killed vaccine. It also leads us to some conclusions that if there is a concern for illness within the calf in that early stages of weaning, modified live may be the best method of prevention by being able to create that greater production of antibodies. And we can conclude that if handled appropriately, using those storage methods and within that 2-hour viability time period for the vaccine, modified lives may provide the greater response in our weaning process. And I would like to thank Sigma Zeta, Southwest Baptist University, John Murphy, and all my fellow researchers for being able to make this research happen. Thank you.